In the Q&A, I talked about the Wigginton article, and with respect to homework 5, some tips on how to compute the p-value in part B and this notation stuff. Basically, in order to compute the p-value in part B, step 1 is to translate your data for SNP number 1 into notation that matches the Wigginton article. So what's in sub A? What's in sub A B? In sub A, and all that. Figure all that out, and then according to the Hardy-Weinberg equilibrium equation, which is, we talked about in the Q&A. I'm going to keep moving, so watch the Q&A for that. There's an equation that describes a level of equilibrium where things are, genes are being shuffled around among progeny in a predictable way. Okay, so I'm going to keep moving. There is another paper Mitchell had asked about next-gen sequencing and SNP calling. So I found a paper that I posted. It's this Nielsen 11 paper. And I'll just briefly make a comment about how next-gen sequencing is used for getting SNP genotypes. So here's the paper, and what I was going to show you was some of the comments on the statistical side. So with next-gen sequencing, how do we decide what the genotype for a SNP is? Next-gen sequencing, we just have these reads. We're not actually able to go and sequence a single base. You can't do that with next-gen sequencing. Next-gen sequencing, you just throw in all the DNA or the RNA. It chops it up, gives you reads. You can align those back to the reference genome, and then you have to decide after the alignment what's the genotype at each of my SNPs, whatever they are. So there are statistical approaches for doing this. The ideal way, okay, so they have a nice picture here. 3 genotype callers. Let me remember what these are. So you can look SNP by SNP and make a genotype call. That's the blue curve. There's a method for doing that. They're referring to the GATK Unified Genotyper as 1-by-1 genotype calls. If you consider all of them together, you increase the call accuracy pretty substantially. And if you use a sophisticated statistical algorithm that incorporates linkage disequilibrium into the genotype calling process, you do much better. So the algorithm that you use for genotype calling, if you're doing next-gen sequencing, is very important. Beagle is an algorithm from the University of Washington. So I don't think these algorithms are available to you in R. These are standalone software packages. Typically, R is where you go after you've done some processing, because these next-gen sequencing data sets, for example, are enormous. The raw data that come out, they're multiple gigabytes of data, and R is not the ideal place to go read in enormous data sets. So if it were me, what I might do, well, actually, if it were me, what I would do is my collaborator in the sequencing facility would do the actual experiment, export the data, and use some of these standalone tools to process the data for me. Ideally, I want the M by N matrix of read counts. That's where is most natural for me to start, because I can go into R and do that. So one other thing I was going to mention was likelihoods. So genotype likelihoods is kind of the state of the art, where we use a likelihood model to say the probability of seeing read counts like that we saw, our read counts, given that the genotype at this location is such and such. If you can come up with a probability model to say the probability that you would see such and such sequence if the genotype at this location was G, then if you had one more component, you could do something pretty slick. So if you have a likelihood and you're willing to propose a separate probability distribution that assigns probabilities to all the possible genotypes at this location. That's called a prior distribution, because this is a distribution of genotypes it does not depend on the data. So before you even look at the data, can you tell me a reasonable prior probability for the different genotypes at each location? When you have a likelihood and a prior, you can apply Bayes' rule and swap the ordering of conditioning. Likelihood is distribution of data given the parameter. Prior is probability of parameter. Posterior is probability of parameter value given the data. Usually when we do statistics, we do what's called what you're used to mostly, I think, is frequentist statistics, which is distinguished from Bayesian statistics. These are two kind of paradigms of doing statistics. Frequentist statistics, it's like there's a population, and in the population there's some parameter that you wish you knew, like in this case genotype, or mean survival time for people with stage 5 prostate cancer. And you treat that as a fixed, unknown quantity. What is the mean survival time of people with stage 5 prostate cancer? If we were able to get every single person in that camp, we could just compute it, and we wouldn't need statistics. Frequentist statistics says that's an unknown parameter. We're stuck with a sample, which is an incomplete picture of the whole population. We want to estimate the parameter, confidence intervals, p-values. That's all frequentist statistics. Bayesian statistics is pretty different, because it treats the parameter as itself a random quantity. It gives the parameter a distribution. Like the mean survival time, we might say so mean survival time would get a distribution. We might say normal at the average survival time that has been observed for the past 10 years for people with that disease. Normal with some standard deviation and some mean. Once we have those two pieces, again, we can write down posterior probability. Now we have built a model, and we can say for this particular location in the DNA, the probability that the genotype is such and such, conditional on the data, is so and so. That's kind of the state of the art for doing SNP calling. Not just binary, are you this genotype or are you that genotype, but estimating the probabilities of the different genotypes. That's how you get big benefits. So when you use a statistical model that incorporates some features of the situation. Bayesian models are very flexible. They can easily accommodate, say, patterns of LD in your data, covariates, other things. All right, that's all I'm going to say about SNP calling, but check out this paper if you want more. And what I was going to start on next was our next topic, which is going to be statistical learning in the context of omics data. So when I say statistical learning, I pretty much mean machine learning or predictive modeling, or discriminant analysis, classification, prediction. All of those have a lot in common. And we're going to use some slides. Some of you have already seen these if you took 636 with me last semester, but they come from Introduction to Statistical Learning. So this book is available free right here. I just Googled the book and went to the first author's website at Berkeley, I think. This is Gareth James, and you can download the book for free in color. There is also, we're not going to use the book, we're going to use some slides. So the authors, Trevor Hasty, and Rob Tibshirani. Rob Tibshirani was the advisor of my advisor, and these guys are they wrote the book on statistical learning. So their first book was called The Elements of Statistical Learning, which became kind of the Bible for this topic, but it's a little bit heavy, not easy to pick up and read if you don't have a PhD in statistics, say. Their second book, Introduction to Statistical Learning, is the sweet spot for accessibility, and it's all in R. So it's lots of code, lots of examples with R. It's not specific to bioinformatics, but they have a lot of bioinformatics examples in there. So context, a little more about the context here. We are still, the goal here is to predict. So specifically, we might want to predict a continuous number, a numerical variable like PSA count for an individual based on their genetic profile. Or we might want to predict a categorical variable like, do you have cancer or not? Give me a blood sample, we'll do an omics technology to it, extract a profile of gene expression maybe, and I want to predict, yes or no, do you have cancer? Or I want to predict, do you have cancer class A, B, or C? Those are both learning kinds of applications. One more thing before we look at the slides, there's two flavors of learning. In this book, they will discuss two flavors of learning, one called supervised, one called unsupervised. Supervised learning is the bulk of the book. That's what you will typically think about when people say statistical learning, machine learning, and that's characterized by cases where you want to predict a value, and you have a data set, a training data set, for which you know that value. So in our context, it might be 20 microarrays from 20 people, 10 of whom we know have cancer and 10 don't. Those are training data because I want to predict cancer status. So for these data, I know cancer status, which means I can build a model to say, this is what the cancer people look like in terms of these profiles. That's what the healthy people look like. Now, when some new observation presents itself for which we don't know cancer status, we apply our model and say, is this profile more like the cancer profiles or more like the normal profiles, and make a prediction. All right, I am going to download the slides. So organized by chapter in the book. We're not going to go through all of this. Today, with the time we have, I'm going to go through chapter 1, which gives an introduction to the problem of doing prediction. Chapter 2 is good. It's overview of some common issues. Linear regression, I'm going to skip. We've talked some about regression, but I'm going to let you guys read more on that if you're interested. We will talk about classification for sure. So classification would be predicting with a categorical variable. That's also called classification. I want to classify you as either cancer or normal based on your input data. We will talk about resampling methods. So whenever you do prediction or classification, you want to be able to say how confident you are in your classification or your prediction. So here's my short little R program. If you take say, a microarray on an individual and plug in their 15,000 numbers from their microarray into my R function, I will tell you a prediction. And I need to be able to tell you, in order to convince you to use that function, I believe this function will make the correct classification 92% of the time. You need to be able to know how accurate the classifier is. Because I could always classify cancer or normal by, say, flipping a coin. If it comes up heads, I'm going to say you have cancer. That's a classifier. It's a poor classifier. So I would need to be able to say, don't expect that to work very often. 50% of the time, you can expect that to work. So hopefully I have my R function that codes up one of these methods we're going to learn and be able to say this is right 92% of the time. If you flip a coin, if you just guess 50% of the time, I can do 90% of the time. So this is a good predictive model. All right, so how do I get that 92%? How do I get that estimate is an important topic, and we will spend some time on it. Might mention this, linear model selection and regularization. Sounds complicated. The idea here in Chapter 6 is we want to do variable selection. So we might have, in our case, we have thousands of variables. We have individuals in our columns, and then we have all these, quote, variables in the rows of our data set corresponding to genes or SNPs or proteins. And I want to predict class membership in terms of all those variables. Especially in the omics applications, we have many more variables than we do observations. We have thousands of genes and dozens of samples, maybe. So that presents problems. If I wanted to, for example, fit, let's say we have data for which yi is 1 if individual I is cancer, and 0 if not. And suppose that for each of our individuals, we have xi, which is the vector of, could be gene expression microarray intensities, or it could be read counts, or whatever, the number for each feature. Individual I, feature / gene 1, individual I, feature 2, feature M. So a simple way to relate cancer membership to input variables would be logistic regression, say, like we talked about in the context of GWAS. We could say log probability individual I has cancer divided by 1 minus probability they have cancer, so the log odds, is equal to intercept. And then we could do this. We could do beta 1 times x I1, the gene 1 effect on cancer membership. Beta 2, the gene 2 effect. There's a model, a predictive model, that could be used to classify. The problem with this model is, suppose we have 20 columns, 10 cancer, 10 normal, and we have 20,000 genes. If you went to R and tried to fit a model that had n equal to 20, let's suppose n is equal to 20 and m is equal to 20,000, if you tried to fit that model, R would complain. You have too many, you can't estimate that many parameters very well with just 20 observations. So long story short, we may have too many variables. And probably not all of them are relevant to cancer versus normal. So variable selection is kind of like what we did with that differential expression stuff. probably not all of these are of interest. Let's do a hypothesis test gene by gene and ask which of them are statistically significant, differentially expressed, interesting. In the context of fitting a predictive model like this, variable selection looks at all those covariates and tries to pick a subset of them. That's chapter 6 stuff. One way to do that, by the way, just to kind of whet your appetite, is principal components. So suppose we have these thousands of features. What's a way I can take lots of variables and boil them down to fewer variables in such a way that I preserve the story or the information or the structure in those original variables? That's something that PCA does. PCA would allow me to take all those M variables and boil them down to, say, a new variable called a principal component, first principal component, second principal component, ... We would have M principal components, but these have been constructed so that the earlier ones explain the most. They are the most informative of all of these. So suppose, for example, we have 20,000 genes, but when we do principal components, we get 20,000 principal components, and if you just take the first 100 features, you explain 80% of the variance, maybe. Then you could probably get away with just putting those top 100 features in the model. And chances are you'll actually do better in terms of accuracy. Suppose that you just made up a variable, or one of the genes on the microarray was totally irrelevant to cancer. Suppose that gene is that one. Do you think it's going to help to put X2 in the model? It's not going to help because it's irrelevant. So if you put something irrelevant into the model, it's actually going to hurt your performance because it's just going to add some extra uncertainty to the model. And it's one extra parameter that has to be estimated. So if it's not relevant, you don't want it in the model. Variable selection is the task of trying to find which ones are relevant. I won't talk about this chapter, but it is basically saying you could do linear regression, you could do even this logistic regression. These are linear. This is a linear model in the sense that this is a linear combination of your covariates. It's a constant times covariate 1 plus a constant times covariate 2 and ... There are more flexible models. So you could have, instead of this being a linear relationship, you could have a quadratic, or you can get very flexible. So to the point where this could be, instead of, this could actually be a function, a smooth function. There's a whole toolkit of statistical techniques for doing that. We will briefly talk about tree-based methods. So maybe you've heard of these, like a random forest, or a random forest and decision trees, boosting, bagging. These are all kind of jargon that's relevant to this type of classifier or predictor. It can be a prediction method as well. Support vector machines are a sophisticated computational algorithm that usually are very powerful. We will talk about them pretty briefly. You can do SVM in R. And then the last chapter. Okay, so I started talking through this set of slides by distinguishing between supervised and unsupervised learning. Supervised learning is what we've been talking about. I have some variable of interest, class membership maybe, that is known for a data set that we have in hand, training data, for which the answer is known. Unsupervised learning is when there is no known answer. There's no grouping that's known beforehand. It's not cancer versus normal. Instead, it's a discovery exercise. So I want to learn about grouping, but I don't have any training data. I don't know what the groups are. So when you see unsupervised learning, basically think cluster analysis and principal component analysis. These are just ways to explore multivariate data in the absence of having a known class. So I'm not going to talk about this chapter, but you could look at it to bone up on PCA and clustering. This has good stuff on those two topics. Okay, where are the slides? Slides. All right. Here on slide 5, there's a few examples of what statistical learning might look like. Risk factors for prostate cancer. So identify variables that associate with prostate cancer. They have some example data. These data, all of their data and all their examples can be accessed in R. There's a package that I think we'll eventually download from Bioconductor that has all their data sets and everything in it. So it's really easy to reproduce what they've done. Let me show you just a couple of examples. So prediction. Predict whether somebody will have a heart attack on the basis of input variables, demographic variables, diet, clinic measurements. Is an email spam or not, based on input variables? Input variables in this case might be keywords, lots of exclamation points, for example, associated with it being spam and not valid email. Image recognition. Here's one that is relevant to us. Classify a tissue sample into one of several cancer classes based on a profile of expression numbers. So here is a picture of a gene expression microarray. All those dots, all those colors are individual dots where there's a gene, and the fluorescent intensity, the redness or the greenness, speaks to abundance. This is a 2-channel microarray, by the way. We talked about that briefly, where maybe the control sample is put in and labeled green, and the treatment sample is put in on the same array and labeled red. So in one array, you get kind of 2 pieces of information. All right, here is a breakdown of the general task. We have some variable of interest, y, cancer yes or no, PSA level, could be numerical, could be categorical. And then we have some predictor variables, demographics, stuff like that. In our case, the predictor variables are genes or features generally, SNPs, genes, proteins. We're either going to do, quote, regression, in which case y is quantitative. So they call it regression. That's just linear modeling, usually linear modeling, where y is a numerical variable. Classification is the same thing, except y is a categorical variable. And then the important piece, in order to do any of this, we have to have training data. So you can't build a smart prediction algorithm if somebody doesn't first show you examples of each possibility. You can't tell the difference between them if you haven't seen some that you know come from each class. Training data would be all the covariates and the outcome measurement for each of n individuals. We want to use the training data to build a model so that we can make predictions on new test cases for which we don't know the response value. Ideally, you would like to be able to interpret the results of your model. So I ideally would not just make a prediction, but I would also be able to look at the results of the model and say, yeah, this is an accurate model, and by the way, what seems to really be driving this model is gene 12 and gene 92, maybe, or gender and age. So some of these methods are more interpretable than others. And I guess a general comment would be that the more complicated the method is, like neural networks and support vector machines are really complex methods. The more complex, the less interpretable it is. I'll say more about that in a little bit. There are very simple methods whose assumptions almost certainly don't hold, but nevertheless often perform well in big data sets and have the extra benefit of being much more interpretable than something like a support vector machine. Let's see. So I want to ideally be able to interpret, and then the last bullet point I said earlier, definitely need to be able to assign some kind of confidence measure to the method. This is my prediction algorithm, and I claim it'll be right 92% of the time. Did you guys hear of the Netflix prize? Netflix prize was 10 years ago, I guess now, but it was data from Netflix. Might as well be omics data. It's not bioinformatics, but it's multivariate data. And for each of many thousand customers, they had training data, which consisted of reviews by customers for different movies on Netflix. And then the goal of the challenge, an open challenge, anybody could have participated in this, download our data from netflix.com, develop a predictive model, send us your predictions on a new data set that we won't tell you the answer for. So here's some training data, thousands of Netflix customers, and for each one, here's the rating on a bunch of movies. So you know the answer, you know the rating on the movies. Now here's a new data set of prior viewing histories, but now predict what their rating will be on these movies that they watched. But you don't know those ratings yet. Predict. And if you can increase our algorithm, this is what Netflix said, if you can increase our algorithm by 10%, they want to be able to make accurate recommendations based on your viewing history. You probably like such and such. Is that a correct recommendation, or are you wasting my time? If you can increase the algorithm by 10%, you win $1 million. In my house, we have me, my wife, 3 kids. So and we all watch Netflix. So some of my recommendations are like Teletubbies. This is kind of interesting. You've heard probably machine learning. These guys are using the term statistical learning. They both mostly mean the same thing. It has to do with who's doing the talking. So if it's a computer scientist, they would probably call it machine learning. If it's a statistician, like Tip Shirani say, who created this stuff, it would be called statistical learning. But in either case, we're just doing predictive modeling for the most part. Okay, any questions about that stuff so far? To finish, let me just show you an illustration of what we're talking about. One of our data sets on eCampus is under microarrays. Let's get the con data. And let's get the con script. So under scripts, data specific, con.r. All right. So the data are microarray data. They come from individuals who have a particular type of tumor, small, round, blue cell tumors. And there are 4 types of tumor: neuroblastoma, rhabdomyosarcoma, and 2 others. So we would like, in this case, to predict, based on your gene expression profile, give me a tissue sample or something and I'll run a microarray. I get thousands of expression numbers for your gene activity profile. Now let's predict. Are you that type of SRBCT, or that one, or that one, or that one? Let's load in the data, and then I'll show you an example prediction. So I need to be in data, microarrays, I think. Data, nope, just con. So CSV, just data. Okay. Go. The first chunk of code is just prettying up the data a little bit and customizing the column names so they're a little easier to read. We have 2,300 genes and 83 individuals. Here is the first part of the actual data file. And what the column names mean, this right here is the first individual in class 2. So back in the script file, I have 1 corresponding to non-Hodgkin's lymphoma, 2 to EWS, and so on. First individual was from group 2, class 2. So here's all the individuals who were in class 2. Then here's some individuals in class 4, followed by some class 3 and some class 1, and then it's mixed up a little bit at the end. Go ahead and make a picture. We'll make that one. I'm not going to make that one. Not going to make that one. Let's do clustering. Let's cluster the samples. I'm going to do hierarchical clustering first. There's the clustering picture, and in terms of making an interpretation of it, looking at the scree plot, I would definitely feel comfortable saying there is at least 2 clusters based on that kind of big jump there. If there were 2 clusters, that corresponds to these over here and everybody else. If we move down a couple more, maybe we feel okay with that being real. If we did do that, we would have 4 groups. That's kind of attractive, since we know we have 4 groups. So we could look at that. So if we broke it into 4, we would have 1. How do I put the boxes on there? Is it like cut tree? Is that what it is? Cut tree. Cuts a tree. Desired number of groups. So if I say cut tree k equal, I think it will make it pretty. Okay, so it just gave me the cluster memberships. So if we said, let's let there be 4 clusters, wonder how that looks. It would be nice if the 4 clusters were the 4 tumor types. That would be, some exploratory evidence that we have good informative discriminatory information for making this call. So let's check that real quick. What I'm going to do to check that, we could pull out, well, all of these 2_1_2 and so on. Those are class 2, and were they grouped together? They didn't have to go into cluster 2. The cluster numbers are arbitrary. I guess what I want to look at is, did the 2s tend to go together? And roughly speaking, I would say no, because there's nearly even breakdown between 1, 2, and 3. There are a few more 2s down here, but most of them are right there. So it didn't look like we did all that wonderfully in terms of classifying group 2. Or it doesn't look like we're going to do very well based on that picture. What about the 4s? Did they tend to go together? Again, not so convincingly. There's a lot of 3s, a few 1s, a couple of 2s. What about the 3s? Here's some 3s. Maybe a little bit better. I see a lot of 2s. Okay, so just a quick picture, a quick peek at the data. Let's go ahead and do PCA since we know how to do that now. Let's do PCA is PR comp data. We're going to have as many PCs as we had samples. FYI. The first PC explains more than half of the variation. So with a whopping 83 variables in principal components, if there were absolutely no structure, I would expect every one of them to roughly explain 1%, less than 2%, if there was nothing. There's obviously something, because there is a principal component that explains all over half. Let's look at the definition of that first principal component. That's going to be hard to do. Let's see. Let's look at the coefficients that define that first principal component. That's hard to interpret. There's too many numbers. But we could... Those all look pretty similar, actually. Do they not? They're all negative, and they're all roughly close to minus 0.1. When we see an index-like variable being the first principal component that explains a lot, that's indicative of there being a lot of correlation in the data. We've seen this multiple times. PCA, we do PCA, we get evidence of something substantial structurally. We look at the coefficients of the first eigenvector. These are the coefficients that would be used to create the first PC, and we try to see whether it tells us anything. Is it a contrast between some features and other features? Are there some features that are driving this and others that are not involved much at all because their coefficients are close to 0? In this case, everybody's contributing roughly to the same extent with the same sign. So if you were to apply this as a linear combination, you would basically have an average of all the gene numbers. So I bet if we looked at the correlation, that's going to be too big. The correlation matrix is going to be 80-something by 80-something, is probably going to have some big numbers in it. Down at line, I think I added a couple of lines, but around line 180, we do classification, so statistical learning. And what I'm going to do is set aside some of the data to use for training, and the rest of it I'm going to hold on to it and not use it for training. We're going to talk about why you do that. We're going to talk about that later. But for now, I'm just going to pick a subset of the data, and we're going to set aside a training data set and a testing data set. Training is what I'm going to use to actually build a model. Testing is what I'm going to use to evaluate the model. So to get the accuracy, the confidence measure for this prediction method, I'm going to use the testing data. We're going to learn a bunch of different prediction or classification methods. One, the simplest, tie for the simplest is naive Bayes. Tie for the simplest is k nearest neighbors. And then we'll see some more sophisticated ones like logistic regression with a statistical technique called lasso. That's a feature selection algorithm. We have too many features. If we were to do logistic or some kind of regression, like a multinomial regression, we have 2,300 features. So I'd have 2,300 beta coefficients in a model that I tried to use to predict. Chances are I don't need all of those. So feature selection, of which lasso is one type, can be used. Let's just use 30 features. Pick the 30 best features in terms of discriminatory information, the 30 features that make it easiest to classify. I'll show you a classification tree. So if you want to start looking ahead at what some of these look like, you can look through, say, the con script. I do the most classification in that script, I think. We'll see a random forest. That's a decision tree. We'll see support vector machines. So just for one example, let's do naive Bayes. Naive Bayes is implemented in this package, E1071. I don't know what that means, but it has several predictive models in it. And several of these that we're going to use operate pretty similarly. You fit a model. So like a few days ago, we did GLM to do logistic regression. We did GLM response tilde gender plus race plus so on. That's how you write a model in R. You say response tilde, as in model the response in terms of some predictors. And we might put here gender plus age plus and so on. When we say dot, that means go to this data frame. That's the column that contains the response. And I want to include everything else as a covariate. We have 2,300 covariates. So this is going to be a model with one for every one. That fits the model. That's like what we did with GLM or like what you would do with LM. To do the classification for a new individual, you could then use the predict function. Predict is a generic method in R. That means that it does different things depending on what type of object you give it. If you did LM and you asked for predict for an LM object, there would be a predict method that was specific to LM. But if you just said predict, R knows how to figure it out. Here, there's a predict method that's specific to naive Bayes. There's a predict method that's specific to most of these. I'll show you them later. So let's do this, and then we'll finish. I'm just going to do naive Bayes, do a predictive model, a classification model, where we use all of the 2,300 features, then use that model to predict on the test data. The test data were not used in fitting the model in the first place. So let's do that first, and then we'll be done in 30 seconds. Maybe not. I'll show you that next time. I don't want to waste time. What we're going to do is get predictions, so 1, 2, 3s, and 4s. For each of the individuals in the test data, here's a 1 or a 2 or a 3 or a 4. That means class, which class was predicted. So then we could compare. For those test data, we do know the answer. We're just pretending like we don't because I want to evaluate my accuracy. So what we could do is just compare, cross-tabulate class membership that was predicted by true class membership. And we get what's called a confusion matrix that tells us accuracies and misclassification frequencies. Okay, we are out of time. We're going to talk about this for several lectures, prediction, classification, statistical learning. After we do this, we will finish up the semester with protein mass spectrometry. We'll do 3 or 4 lectures on mass spec. Questions? All right, have a good weekend.